Hello friends, we have started discussing about bacterial plasmids. In the first part of this video, we discuss about introduction of bacterial plasmids. Then we discuss about some of the facts and terms related to plasmids. We also discuss about characteristics and properties and also role of plasmids. And finally, we discuss about general structure or elements of plasmid as vector. You can refer my video, the link is given in the description box. In this video, we are going to discuss about types of plasmids. So first of all, let us see the types of plasmids on the basis of their ability to transfer to other bacteria. On this basis, these are of two types. First is conjugative plasmids. These code for functions that promote transfer of the plasmid from one bacterium, that is the donor bacterium, to the other bacterium known as the recipient bacterium. So what we can say that these are capable of horizontal gene transmission. Thus, they perform conjugation. These have tragenes that help in the process of conjugation. Example, synthesis of sex pili, which is then later converted into conjugation tube and helps in the transfer of plasmids from donor to recipient. This type of plasmids are found abundant in naturally isolated bacteria. Large plasmids whose size is greater than 40 kilobase pair are often conjugative plasmids. Second is non-conjugative plasmid. These are incapable of initiating conjugation because they do not encode self-conjugative system. Hence, these may be transferred from donor to recipient bacterium in assistance with conjugative plasmids. Plasmids smaller than 7.5 kilobase pair are often non-conjugative plasmids. Now, types of plasmids based on function. On the basis of this, these are of 5 types. First is fertility, in short F plasmid. These are also known as fertility or sex factor. These determine the sexuality of bacterial cell. Bacterial cell in which F plasmid is present is known as F plus male or donor cell. And bacterial cell in which it is absent is known as F minus, that is female or recipient cell. These are transferred from donor to recipient through conjugation. Hence, what we can say that these are conjugative plasmid. These exist independently or integrate into main bacterial genome or chromosome. Therefore, F factor is an example of episome. A bacterial cell with an integrated F factor is known as HFR cell, that is high frequency recombinant. Functions of F plasmid carry genes, that is tragenes, to promote conjugation, that is transmission of F plasmid from donor to recipient cell. Second is resistance, or in short R plasmid. These confer resistance to one or several antibiotics. Some R plasmids have a single resistance gene. Then host cell is resistance to one antibiotics. And if R plasmids have multiple resistance gene, then host cell is resistance to several antibiotics. Hence, these are threat to medical sciences because if the bacterial infection is caused by bacterial cell in which R plasmid are present, then these are resistant to antibiotics. Hence, this type of infection cannot be treated by that particular antibiotic. R plasmids are conjugative plasmid. It also provides immunity to harmful effects of heavy metals to host cells. Now, the third type of plasmid is Col Plasmids. Col Plasmids contain genes that produce bacteriocins. So now what are bacteriocins? Bacteriocins are antibacterial polypeptides which kill different strains of bacteria. And how they kill bacteria? By inhibiting one or more essential process as DNA replication, transcription, translation or energy metabolism. Bacteriocins that are produced by E. coli are known as colicins. Colicins inhibit the growth of susceptible bacteria, or in other words, sensitive bacteria. There are many types of colicins produced by different strains of E. coli, and these are designated by a letter. Example, colicin B, etc. And each inhibit the growth of bacteria by particular mode of inhibition. Fourth is degradative plasmids. Degradative plasmids helps the host bacterium to catabolize organic compounds that are not commonly found in nature, as xylene, toluene, camphor, etc. There are certain genes present in these plasmids that encode enzymes which degrade these compounds to produce carbon and energy sources. These are also conjugative plasmids. Degradative plasmids increases the chance of survival of host cell in the presence of these unique substances or in the presence of these unique conditions. Now fifth is virulence plasmids. Virulence plasmids can give pathogenicity to host bacteria, or in other words, turns that bacterium into a pathogen. What are pathogen? Pathogens are agent of disease or disease causing agents. Disease causing bacteria easily spread and replicate among affected individuals. Example, E. coli has several virulence plasmids. Similarly, Salmonella enterica also has virulence plasmids. While discussing F plasmid, we came across episome term. What are episome? Plasmids having the ability to integrate in the chromosomal DNA of host cell are called episome. So, plasmids which are capable of incorporating into the main bacterial genome or bacterial chromosome are known as episome. It remains integrated for prolonged periods and are replicated along with host chromosome during cell division. Eventually, it becomes essential part of host genome. Example, F factor. This F factor we have already discussed. A bacterial cell with an integrated F factor is referred to as an HFR cell. Full form, high frequency recombinant cell. This is because of high efficiency for chromosomal gene transfer. So, in HFR cell, chromosomal genes are transferred from donor to recipient with high efficiency. This we will discuss in detail in bacterial conjugation topic. So this is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.